Welcome back to the deep dive. Glad to have you with us again as we try to make sense of the complex stuff. Today we're diving into something really quite fascinating inside your body, how it manages energy. Specifically, this process called glycogenolysis. And our mission really is to get our heads around this neat bit of biological specialization. How two major players, your liver and your muscles, both store the same fuel glycogen, but then break it down for, well, totally different reasons. We're going to unpack the tiny molecular switch that decides if that energy goes body-wide or stays super local. And for this, we've got some great insights from a YouTube video, Glycogenolysis, Liver versus Muscle at Metabolism Made Easy. Pretty clear title. By the end, you should have a much clearer picture of why your liver is like the main glucose handout center, while your muscles are a bit more focused on themselves. It's really quite elegant when you see it. Let's jump in.
Absolutely. And what we're digging into today is, well, it's fundamental if you want to understand your body's whole energy setup. Glycogenolysis, it sounds complicated, but at its core, it's just breaking down glycogen. And glycogen is just the stored version of glucose, right? Think of it like a big branch chain of glucose molecules packed away, ready for quick access. But the really cool part, the aha moment, is how breaking down the same stuff in the liver versus the muscle leads to, well, completely different outcomes for the body.
Okay, so glycogen is the reserve tank for glucose. We eat carbs, they become glucose, and any extra gets stored away as glycogen, mostly in the liver and muscles. Makes sense. Keeps our blood sugar stable, gives us energy for moving around. But yeah, you hinted that where it gets broken down changes everything. That's the puzzle.
Exactly. And to really nail that difference, we need to talk about one key molecule first. It's called glucose 6-phosphate, G6P for short. See, when glycogen breaks down in either tissue, liver, or muscle, you first get glucose 1-phosphate, which quickly becomes this glucose 6-phosphate. Now, the important thing here is that phosphate group stuck on the glucose. It effectively traps it.
Traps it? How so?
Well, it gives the molecule a negative charge and makes it too bulky to just slip through the cell membrane. So it's basically locked inside the cell way it was made.
Ah, okay. So glucose 6-phosphate isn't free glucose yet. It's like earmarked energy stuck inside the cell. That makes sense. You wouldn't want it just leaking out everywhere. So let's follow that G6P. What happens in the liver? You said its role is different, more for the whole body.
Precisely. The liver's job with glycogenolysis is, you could almost say, altruistic. It's not just feeding itself, it's managing blood glucose for everyone else, essentially. So when your blood sugar dips, maybe you haven't eaten for a while, the liver gets the signal, okay, time to break down some glycogen. And that process makes this trapped glucose 6-phosphate.
Right, so the liver's full of this trapped G6P. But how does it become the glucose dispenser you mentioned, especially for like the brain, which needs glucose constantly? How does it get the glucose out?
And that is where the magic happens. It comes down to one special enzyme that the liver has. It's called glucose 6-phosphatase.
Glucose 6-phosphatase.
Okay. This enzyme is like a key. Its specific job is to snip off that phosphate group from glucose 6-phosphate. Once that phosphate is gone, you're left with just plain old glucose, free glucose.
And free glucose isn't trapped anymore.
Exactly. Without the phosphate, it loses that charge, it's smaller, and it can easily pass through the liver cell membrane and into the bloodstream, ready to travel anywhere in the body.
Wow, okay. So that one enzyme, glucose 6-phosphatase, is the absolute difference maker. Without it, the liver's glycogen stores would be useless for raising blood sugar systemically.
You got it. That enzyme is everything for the liver's role here. It's what allows the liver, and actually the kidney to some extent too, to export glucose. Most other cells just can't do that.
It really highlights the liver's central command role in metabolism, doesn't it?
Absolutely. It's the gatekeeper for blood glucose.
Okay, that paints a really clear picture of the liver acting for the greater good. Now let's flip to the muscle. What's its story with glycogen and that trapped glucose 6-phosphate?
Right, so muscle tissue. It also stores a lot of glycogen, maybe even more than the liver in total body mass. But it's all earmarked for one thing, fueling muscle contraction. When muscles break down their glycogen, they also make glucose 6-phosphate, just like the liver. But, and this is the crucial point, muscle cells do not have glucose 6-phosphatase. They lack that unlocking enzyme.
Ah, so no phosphatase means
Right.
the phosphate stays on.
Stays on, which means the glucose 6-phosphate remains trapped inside that specific muscle cell.
And if it's trapped, it can't get into the bloodstream. So muscle glycogen can never be used to raise overall blood sugar levels.
Never. It's physically impossible because it can't be converted to free glucose to leave the cell.
So what does the muscle do with all that trapped G6P then?
It uses it immediately. That glucose 6-phosphate gets fed directly into the energy-producing pathway within that same muscle cell, the pathway called glycolysis. Glycolysis rapidly generates ATP, which is the direct fuel muscles need to contract, whether you're lifting weights, running, or, just standing up. It's a very efficient closed-loop system for local power.
Okay, so the muscle is like a self-contained power station. It stores fuel, breaks it down, and uses the energy right there on the spot. Completely different strategy than the liver.
Totally different. It's a beautiful example of specialization. The body ensures the brain gets its steady glucose supply via the liver.
While the muscles have their own private stash ready for immediate action without dipping into the main supply needed elsewhere.
Exactly. It prevents competition, in a way. Each tissue is optimized for its primary function.
That basic difference is clear now. Liver shares, muscle keeps. But how does this play out in real-life situations? Like, say, fasting versus intense exercise?
Great question. That's where you see the system dynamics. So during fasting, like overnight, your blood glucose starts to fall. This triggers the release of a hormone called glucagon from your pancreas. Glucagon's main target is the liver. It tells the liver, hey, break down glycogen. And importantly, it also activates that key enzyme, glucose 6-phosphatase.
So glucagon specifically tells the liver to release glucose into the blood.
Precisely. The liver then pumps out free glucose, keeping your blood sugar levels stable, especially for the brain, until you eat or other processes like gluconeogenesis kick in more heavily.
Okay, liver handles the fasting response. What about exercise? When muscles are screaming for energy?
During intense exercise, the signals are different. You get adrenaline, sure, which can stimulate glycogen breakdown in muscle, but also just the sheer demand for ATP within the muscle cell triggers local glycogenolysis. That glucose 6-phosphate, remember, stays inside the muscle. It doesn't need glucagon. It doesn't care about blood sugar levels. It just gets funneled straight into glycolysis to make ATP right then and there.
Super fast, on-demand energy, exactly where it's needed, without bothering the rest of the system.
That's the beauty of it. It allows the liver to focus on maintaining systemic glucose while muscles power themselves independently. True metabolic teamwork.
This is so elegantly designed, it makes you wonder, does it ever go wrong? Like, are there conditions where that liver enzyme, the phosphatase, is missing or doesn't work?
Yes, absolutely. There's a group of genetic disorders called glycogen storage diseases. And type I, known as von Gierke disease, is exactly that, a deficiency in glucose 6-phosphatase.
Wow. So what happens to people with that?
Well, they can store glycogen in the liver just fine, but they can't release the glucose effectively. So they suffer from severe hypoglycemia, really low blood sugar, especially when fasting. They also tend to get a buildup of glycogen in the liver and kidneys because it can't be broken down properly for release. It really highlights how critical that single enzyme is for health.
That's a stark reminder. A tiny molecular detail with massive life-altering consequences. It really puts the precision of our normal metabolism into perspective.
It really does. Just thinking about how this setup allows us to handle such different states, sitting quietly, fasting for hours, or sprinting flat out, it's all orchestrated by these kinds of specific molecular tools, like having the right enzyme in the right place.
It's pretty amazing. And that, I think, brings us towards the end of this particular deep dive. We started off asking why the liver and muscle handle their energy stores so differently, and the answer really seems to hint almost entirely on one specific enzyme, glucose 6-phosphatase. The liver has it, so it can snip off that phosphate, release free glucose, and manage blood sugar for the whole body. A real team player. Muscles don't have it, so their glucose 6-phosphate stays trapped, gets used immediately for glycolysis, powering just that muscle. Much more self-sufficient, as you said.
Self-sufficient works. And when you zoom out, you see how this cellular difference impacts everything. Your energy levels during exercise, how you cope with fasting, just your overall metabolic health. It really makes you think, doesn't it? This leads to a broader question, perhaps for you listening. How many other tiny differences like this, maybe just one enzyme here or one receptor there, are quietly running massive essential functions in our bodies, functions we probably never even consider? Maybe reflecting on these intricate energy pathways could change how we approach fueling ourselves for different things. Or maybe just gives us a moment to appreciate the incredible, silent, specialized work happening inside us right this second.